Welcome everybody again to another episode of the ideaXme show, the show that profiles the humans behind the really big ideas that are shaping our world and inspiring future creators, and for all those that really like a good story. I'm Ira Pastor. I'm your aging and longevity ambassador along this journey. And for the last few shows, we have been spending time at different hierarchical levels of the biologic architecture of life and aging. We spent time talking about the genome, the micro-biome. We've dabbled a bit in quantum biology and the hydrodynamic aspects of life. However, we can't forget that while all of these integrated and varied biologic processes are important for the maintenance of our daily health, that when things go wrong, the trickle down is unfortunately a wide range of chronic degenerative diseases responsible for human suffering and death, and an annual global healthcare expenditure in 2019 of around $7 trillion. And unfortunately, one of the major set of pathologies responsible for part of that figure in 2019 remains cancer. And despite the major progress that we've made over the last century on improving outcomes and throughout sort of War on Cancer 1, we still have a sobering set of numbers in front of us. The World Health Organization data for 2018, basically 18.1 million new cases of cancer, 9.6 million deaths. So today's guest, who's going to take us further along the theme as to where the future is taking us in the oncology field as we enter War on Cancer 2 and strive to have more clinical impact, is Dr. Dario Altieri, who is the President, Chief Executive Officer, and Director of the Wistar Institute Cancer Center here in Philadelphia. Dr. Altieri got his medical degree at the University of Milan School of Medicine. He earned a postgraduate specialty degree in clinical and experimental hematology. 1987, he joined Scripps Clinic and Research Foundation in La Jolla. 1999, he was named professor of Yale University School of Medicine. In 2002, he became the founding chair of the Department of Cancer Biology at University of Massachusetts Medical School. He joined the Wistar Institute back in 2010 as its chief scientific officer, and he was appointed to the CEO role in 2015. And just as a side note, for anyone unfamiliar with the Wistar Institute who do not have the pleasure of living in the epicenter of the Philadelphia biomedical scene like I do, it is the nation's first independent institution devoted to biomedical research. It was founded all the way back in 1892, and to this day remains a leader in biomedical research in both cancer and infectious diseases, focused on discovery of a wide range of therapies and vaccines. 1972, it was named an NCI Cancer Center and Basic Research, a distinction it holds till this day. And over the years, it has developed numerous vaccines, identified a range of genes associated with cancer, developed monoclonal antibodies, and many other significant research technologies and tools. So with that, welcome Dr. Dario Altieri to the show.
Thank you, Ira. It's a pleasure to be here. I'm flattered to be able to join to participate in your in your show.
Thank you so much. So we usually just open the show with giving our guests the floor. The audience would love to hear about you, where you grew up, how you got interested in science, how you got interested in medicine, and then ultimately what led you into oncology.
Sure. As you already indicated in my bio, I have a medical education, and I train in clinical and experimental hematology as my specialty degree. And I think that my decision to get into medicine and attend medical school was probably one of the things that I did right in my life. Is I never regretted it. I think it's a great profession, it's a great training, and it was an amazing experience. I have to say though that I caught the bug about research and laboratory very early on. Hematology in particular is a very specialized specialty, in the sense that you really need to keep up to date with the research advances. Very different from other disciplines in medicine, like primary care, of course. So where even that you really need to be up to date. But hematology is really a highly specialized discipline. And as part of of my postgraduate training after my medical degree, I started working in the lab as a part-time physician scientist, and it was a lot of fun. It was a lot of fun. It's the journey of discovery, not knowing really what you're gonna find. and the opportunity to ask questions, even the most abstruse ones, and things that might not make sense, has just been a thrill. And now, I've been doing this for a very long time. I'm 60 years old, and the thrill is still there, and the lab is still my top passion. So the second part of your question was, why oncology? And I think, some of the motivation is what you said. We've made great progress, in cancer research and cancer care. There have been remarkable milestones. There's been tremendous progress in the management of certain types of cancer, but there is still a lot of work that needs to be done. And just to add one more number to the ones that you mentioned, in the United States alone, it's expected that over 650,000 people will die of cancer this coming year. And we made great progress. Mortality rates for cancer have declined over the last 25 years, but that's still a staggering number. And in addition to that, there is tremendous hardship that comes attached with the diagnosis of cancer, certainly for the patient, but also the family. There are financial hardships. And so I think that the mission is still there, and one that requires a level of urgency that has remained unabated over the years.
Wonderful. Yeah, I completely agree with you on that front, and I'm glad to see that you're leading the focus there down this path. Looking, more in depth at the activities going on at Wistar. Basically, when you go to the website, you see these three core pillars. So you have sort of the gene expression regulation activities, looking at core biology and gene regulatory networks behind oncogenic transformations. You have activity in oncogenesis, molecular and cellular platform, looking at the cancer signaling and tumor development, and then a sort of dissemination platform involving metastasis, changes in tissue microenvironment, and sort of the connected immunology. And overarching all of that, you have this very potent sort of computational biology structure, which I'm sure is to help you sift through sort of very large amounts of genomics and proteomics, metabolomics data, and so forth. Can you briefly talk about the importance of some of these tools, these computational tools and others, whether they be machine learning, artificial intelligence, or other things that I'm not familiar with, in helping you better understand cancer today and deal with these, what I understand are exceptionally large amounts of data now that you have to deal with?
Absolutely. And that's, you focus the question, I think, very appropriately. And this is, in fact, one of the most significant challenges in modern cancer research. We have gotten very good at the technology. We can sequence the genome of our patients in a very short period of time, no more than 1 or 2 weeks. The cost of doing that has come down dramatically. Used to be the first genome sequencing project was over $1 billion. Now it's affordable. It's something that you put on your credit card. Mm-hmm. And so the technology has progressed tremendously. We've been able to advance the machines and the tools that are needed in order to sequence billions of base pairs of genomic material. But there is a bottleneck, and the bottleneck is data interpretation. Mm-hmm. We're not exactly sure which side is up oftentimes. I mean, when there is cancer is a disease of nucleic acid, it's a disease of our genes. Mm-hmm. How we get to alterations in the DNA is complex, is multifactorial, is due to many different facets. But in essence, we make mistakes. Our cells make mistakes. And they make mistakes because their DNA is altered. Mm-hmm. And normally we have checks and balances, and we have ways to eliminate cells that have made a mistake. Sometimes those fail-safe mechanisms don't work. Right. And that is where we generate a progeny of cells that pass on that mistakes on to their daughters and daughter cells. And so, sometimes it's very straightforward to identify a so-called driver mutation. Mm-hmm. Sometimes it's a change in the DNA that is clearly associated with the disease and is clearly not just a marker, but is really the engine that keeps the tumor going. But unfortunately, this is actually the minority of cancers. The vast majority of cancers have multiple mutations, hundreds of mutations, and sometimes we are in trouble in identifying and clearly assigning which one is the driver and which one is a passenger. Something that comes along that is not really a clear driver of the disease itself. And so the computational aspect has become really the key to interpret billions of base pairs that are being sequenced and try to identify certainly the most common mutations, the most common changes in the DNA, but also the ones that occur in a very small fraction of the cells. And in the past, we've been saying that those probably are minor, they don't count, but in some cases, because every type of cancer is generated by a collection of clones, sometimes mutations that occur even in 2% of the tumor cells are the ones that we need to pay more attention to.
Right.
And that can provide also some vulnerabilities in terms of therapeutics. So Wistar in particular, focuses on basic biomedical cancer research. We try to identify genes and pathways, signals that keep tumor cells alive, and as you were saying in your introduction, enable eventually the dissemination of tumor cells. Ultimately, we need to, in order to conduct modern cancer research, we really need the technological infrastructure to make sense of the genetic makeup and the genetic fingerprint of a tumor. And as you know, what we call cancer is really probably a collection of over 200 different diseases. And what we have learned is that even two patients with seemingly identical types of tumors, in fact, can have at least two completely different diseases.
Absolutely, absolutely. And that theme of heterogeneity is one that you see sort of in this evolving world of sort of systems thinking. You see it more and more, where, as you were saying, the old school view of this homogeneous thing is just not what we have, and you have these masses that are mixtures of many potential different cells, different transcriptional regulatory states, and you remove one, others can reconstitute, and it's this whole concept. And I'm glad to see that you're focusing so much on this. Is really, when you see these amazing sort of stories in the news about a subset of patients with some type of smart therapy that tumors melted away, but it didn't work in everybody else. It's really, I guess, it shines a light on the importance of dealing with this heterogeneity issue as you move forward. And speaking along those lines, so specifically looking at your lab at Wistar, you're very focused on tumor cell survival, mechanisms of cellular adaptation, the different processes that promote resistance, and so forth. And you are interestingly very focused on mitochondria in studying the chaperones and the other moieties that regulate bioenergetics and adaptation. Mitochondria is an extremely interesting topic, not just for its connection to cancer, but obviously the vast array of mitochondrial diseases and aging in general, and being such a important powerhouse of cells, but also, and something you were alluding to, unlike the nuclear genome with its 25,000 or so genes, in that mitochondrial genome, you have what? You have 37 or so genes to the 25,000 or so that you deal with in the nuclear genome. And, there was I read a paper recently about down at NIH in allergy and infectious diseases, how they were looking at heterogeneity in the mitochondrial DNA as a prognostic agent for liver cancer, and having some very good successes in sort of screening tumors and so forth. Can you speak a little bit more sort of to the therapeutic and strategic importance of mitochondria as a target with its, very defined genome versus the much more complex nuclear genome?
Sure. I mean, I think some. Let me take a step back and saying that the way tumor cells get their energy has been a very complex biological journey. Because tumors tend to revert, for the majority of them, not all of them, but they tend to revert to fermentative metabolism where they utilize glucose and even in the presence of oxygen. And of course, the glucose is the fuel, you know, that comes through with the food, and that's what powers up all of our cells. And the issue is that, you know, about 2.5 billion years ago, we happened to encounter a very smart bacteria. And normally bacteria, you know, are bugs, and we fight infections, and you know, now we take antibiotics. But our body is very good at fighting infections. But it happened that that particular bug 2.5 billion years ago was a very smart one, and turned out to be very useful for the eukaryotic cells. Mm-hmm. It was very smart because it could generate a lot more molecules of ATP, which is really the chemical backbone of our energy, of our cellular energy, could generate a lot more molecule of ATP than glycolysis, which is the simple breakdown of glucose that we get through the food. And so, you know, biology keeps what's useful and gets rid of what is not. And so we kept that bacteria around in our cells, and that bacteria turned out to be the mitochondria. Mm-hmm. And so basically our energy production modality utilizes a very complex, fascinatingly complex process in the mitochondria that makes a ton of energy, a ton of ATP per mole of glucose. Tumor cells, for reasons that to this day we're not entirely sure of, go back to the world that was before mitochondria, and rely on glucose breakdown to make ATP. Is inefficient, generate a lot of waste called lactic acid, but for whatever reasons, tumor cells thrive in that environment. And so the role of mitochondria in cancer has been controversial, and to the point where people believe that in cancer, mitochondria must be dysfunctional because tumor cells don't really use them very much. That turned out not to be true. Mitochondria are perfectly functional in cancer. And again, there was an example where we tried to look at cancer in its as if it were an homogeneous, you know, carpet of cells. And in fact, that is not the case at all. We have clones of, think that, you know, when we see a spot in a chest X-ray, 1 centimeter in diameter, 1.5 centimeter in diameter, that's 1 billion cells, 10 to the 9 cells. I mean, it's what that means is that in that billion cells, you have individual clones that are constantly competing and cooperating with each other. And the same argument of interdependence, competition, and cooperation applies to energy production. And so it turns out that different areas of a tumor, even if it's just 1 centimeter in diameter, different areas of the tumor have different tumor cells that utilize mitochondria or glycolysis, either in mutually exclusively or at the same time. Mm-hmm. And so now we have a better understanding that, in fact, not only mitochondria are functional in cancer, they're actually very important in driving very specific cellular traits, cellular characteristics of tumor cells. And one in particular that has emerged in recent years, and that's been a focus of my own lab for quite some time, is in fact the ability of tumor cells to metastasize. Mm-hmm. As you know, Ira, metastasis is the dissemination of tumor cells from a primary tumor to distant organs. And 90% of people who die, unfortunately, die of cancer. In fact, they die of metastasis. They don't die of the primary tumor. Sure. And so it's a huge medical problem. And unfortunately, when we were discussing at the beginning areas where our progress has not been as extraordinary as we would have liked, one of those areas is, in fact, metastasis that is currently considered an incurable condition. Patients, unfortunately, that come at the attention of their physicians with disseminated disease to multiple organs, whether it's the liver, the lung, the bones, unfortunately have a disease that is treatable, but it's not curable. And so it turns out that mitochondria seem to be playing a very important role in that process. Whether it's the only role that they play, certainly not. They are, you know, in addition of being the powerhouse of the cells and regulate energy production, they're also the key organelle that controls cell death and cell survival mechanisms. And tumor cells are very good at eliminating or thwarting mechanism or stimuli that would otherwise kill them. and manage to, in fact, stay alive. And that is actually what we see in the clinic, a very big problem of chemo resistance.
Right.
Patients are being treated for cancer. The good news is that the tumor shrinks. Everybody is happy. There seems to be a clinical response. That clinical response can be durable. And then all of a sudden, the disease comes back. And unfortunately, at that point, is resistant to the therapy. Mm-hmm. And the basis of that mechanism is, in fact, the ability of tumor cells to resist the process called apoptosis, which is the definition of programmed cell death. And that takes place also in the mitochondria. So what we're trying to do, the big question that we're trying to address in the lab is how these processes are interconnected, are intertwined in conferring a level of plasticity to tumor cells that really defines a very high degree of adaptability. Tumor cells adapt very quickly to virtually anything that we can throw at them, whether it is treatment, whether it is chemical treatment, radiation treatment, immunotherapy, all the way down to conditions of the tumor microenvironment that oftentimes are not particularly favorable for tumor growth.
No, that's wonderful, and it's a very important area of research, obviously, for the not just for the the control of that adaptation, but as you said, the metastasis and understanding there's some theories in sort of cancer technically evolutionarily goes all the way back to single cell organisms, and some of those patterning mechanisms and the way single cells used to agglomerate together when the first multicellular organism showed up. So it makes a lot of sense why these organisms prove so damn resistant, and it's good that it's very important that you're working along that particular set of targets. One other thing I wanted to bring up, you're sitting here in Philadelphia and talking about oncology. I have to bring up a paper. It's almost 45 years now. But back in 1975, a rather famous embryologist by the name of Beatrice Mintz at Fox Chase Cancer Center published a paper. It was entitled Normal Genetically Mosaic Mice that were produced from malignant teratocarcinoma cells. And back in 1975, this was kind of earth-shattering. It was sort of the first evidence of this ability of a microenvironment, in this case an embryonic microenvironment, to, for lack of a better word, normalize a phenotype, organize in what it needed, organize out, and you had these mice that were born and they had no cancer and so forth. And this was described over the years, at the time, terminology embryonic fields were used. Similar dynamics were seen in the plant kingdom. You see some things related to this dynamic in regenerative organisms like salamanders and planarians. Regenerative organisms are very difficult to kill with cancer. And the term epigenetic obviously comes along, and nowadays, in the molecular biology sense, epigenetic marks typically embody DNA methylation and histone modifications and so forth. Back then, the term was a much broader sort of epigenetic landscape. The concept of it was coined by developmental biologist Conrad Waddington on how sort of gene regulatory networks can move sort of attractor states, stabilize gene expression patterns across the genome. And where I'm going with this is, whether we're talking about older therapies and radiation and chemo or some of the newer smart therapies, we're always looking at a kill event. Yet, there was a brief period of time, I guess a decade or so ago, where you saw sort of these epigenetic modifiers popping on the scene, not to, I guess, kill a cell, but to reprogram it in vivo and turn it into a normal cell. Where have we gone with this? Because it seems like a path that was hot maybe at the beginning of the 2000s, but you don't hear too much about it lately.
Yeah, it's a fascinating field. It truly is. I think that you're absolutely right that there are histone deacetylase inhibitor that have been around in for a few. Sorry for the phone. that have been around for a few years. And some other treatments for leukemia in particular have been centered on differentiating the leukemia cells. You know, leukemia is a disease of a block of differentiation. We have, we know that cells that are originated in the bone marrow have to go through a number of steps in order to mature and differentiate into the mature cells that do what it is that they do. So neutrophils fight infections, red blood cells carry oxygen, and platelets stop bleeding, hemorrhages. But before they get to that point, they have to go through multiple stages of differentiation. Leukemia is a block, it's due to a genetic alteration, due to one or multiple mutations, is a block of that differentiation steps. So we accumulate a lot of these cells that don't actually do the job that they're supposed to do. Mm-hmm. And some treatment, in fact, the things that, we read in mystery books because it's been used as a poison, like arsenic trioxide, as was part of traditional Chinese medicine for 5,000 years, it turns out to be actually a very active and very significant treatment for the management of specific types of leukemia. Sure. Perhaps not by killing the leukemic cells, which is what chemotherapy does, but by inducing them, by forcing them through the differentiation pathways, by forcing them past the block imposed by the genetic defect. Sure. I would say that, it's still a fascinating area. I would say that it's still a truly interesting approach, and one that would eliminate, if completely successful, would eliminate the risk of recurrence. Because in essence, you eliminate the tumor cell population, not by killing all of them, but in fact, by inducing their differentiation into a progeny that doesn't proliferate anymore. But I think the experience is still limited. And our current therapy, whether it is molecular therapy with kinase inhibitors, or immunotherapy by reawakening the immune system in cancer patients, or the 1960 vintage chemotherapy and radiation, sure. All of those approaches are still centered on the idea of eliminating all of the tumor cells. And I emphasize the word all because in essence, that's what it takes in order to prevent the recurrence. I mean, there are models of leukemia that suggest that even if one cell stays around, that is sufficient to produce a relapse. Mm-hmm. And at that point, the prognosis becomes very unfavorable. Yeah. So it's a fascinating field of investigation, but as and maybe we're going to talk about that in a moment, but currently the real revolution in cancer therapy is immunotherapy. And that has generated tremendous momentum, and the clinical responses are, I think, in some types of tumors, nothing short than exceptional, extraordinary. And that is where a lot of emphasis is now being placed.
Excellent. I appreciate that. Definitely. So before we wrap up, and once again, I'll give you the floor at the end of the show to give your grand vision on where you see things going. But before we get there, I have to segue into one question. I call it the science fiction question of the interview. We usually ask the guest, if there was somebody in history, could be 10 years ago, 1,000 years ago, whenever, that you would have liked to have met and talked to, an artist, a scientist, an explorer, who would that have been?
I have a very clear answer. I don't have to think about that.
Let's hear it.
I would have liked to meet Jonas Salk.
Okay.
Jonas Salk, as you know, is the originator of the vaccine against polio. Mm-hmm. Immediately after World War II, the American public, almost at the height of the Cold War, the American public was more terrified of polio than a nuclear exchange with the Soviets.
Right. I remember.
And this is to emphasize what a social impact that disease really truly was. And you remember the pictures of the kids in the iron lungs. I mean, the worst nightmare that a family could experience. And Salk, he competed with a number of other groups for the Sabin was another one,
Right.
and Hillary Koprowski from Wistar was the third one. The race was on to identify a safe, protective vaccine against polio. And, there were different changes. The biology was not really known at that time, but it turns out that the oral vaccine, this is something that you ingest, was put, a couple of drops were put on a cube of sugar. The oral vaccine was superior to all of the others because it generated a long-lasting protection. Salk was hailed as a world hero, a disease conqueror. And the last thing that I want to mention that I think is very important for me and an inspiration, I would say, is that Salk never patented his vaccine. And it's really unclear how much profit he gave up by not doing that. It is estimated that we are in the order of billions.
Yeah.
And I think that this has to do with culture, character, and the way we have to think about science. Certainly, we live in a world and in a society where everybody has to make money, right? Healthcare is no different.
Right.
Drug discovery and drug development is no different. But it wouldn't be a bad idea to go back to the concept of that science really doesn't belong to anybody. It's not ours. It's everyone's. And that was the message that Salk once gave during an interview when he said, "You can't patent the sun."
Right.
That keeps us all alive. So that's my long speech to say that, I think it was great.
No, I appreciate that. It was very elegant, and I totally, having sat around this industry for the last 30 plus years, I completely can sympathize with that view. This is a side note. I had the honor of meeting Hillary Koprowski several years ago.
Goodness.
Yeah, he was getting up there at the time, but of all things, he was looking to produce recombinant, rabies vaccines and tobacco plants. I was with an agricultural biotech company at the time, so I did have the honor of meeting him a few times. But yeah, amazing set of minds there. So
yeah, I mean, it's not exactly congenial minds. These people would fight ferociously, for credit and for getting there in the first place. But in the end, science is fun, Ira, but it can really help millions.
Oh yeah.
And so that was, one of those fundamental achievements.
Yeah. Yeah. Well, we're coming to the point where we're going to wrap up. Dr. Dario Altieri, President, Chief Executive Officer, and Director of the Wistar Cancer Center. I'll give you the floor. Take the show out. I mean, I'll finish up with your grand vision. The audience would love to hear where you think we're going to be in the next 5 years, the next 10 years, and how we're going to be dealing with this scourge that we call cancer. And
well, it's and this is without dwelling into any politics, Ira, but I think where we're going to be, a lot of it really depends on us. Biomedical research, and particularly cancer research, has to be a publicly funded effort. It has to be outside of market considerations, has to be outside of profits. It just cannot be owned by anyone other than the taxpayer. It's something that I truly believe the United States has always been a leader in innovation and a leader in the freedom to discover. And that freedom to discover is the one that has produced some of these groundbreaking transformational advances in conquering diseases. Progress in cancer has been slower than we had hoped, but has been tangible. And now I think that unfortunately there is a perception that research is a luxury, that we don't need to invest in research, that, you know, it's somebody else's problem in a way. The drug companies can do it, the biotechs can do it. And I would strenuously argue that it just cannot happen. And so where we're going to be as a society really depends on our willingness to continue to make steady commitment towards biomedical and translational research. Many things, most of the things that we do don't work and will not work. Some will work a little bit, one or two will make history. But that can be done only as part of community engagement and a social understanding that only research can provide the answers that eventually will conquer disease, including cancer. And I've been doing this for 35 years, and it truly is not a job, it's truly an adventure, but that can only, and maybe we as a community have not really done a good job in communicating the importance of what we do. And we see this every day, where our message is maybe not as tangible as it should be, or whether we ourselves get ahead of ourselves and we talk about magic bullets when in fact, you know, only to go back to the public, say, well, you know, maybe it really wasn't a magic bullet. But nevertheless, continuity and persistence is really what wins the day in a lab. So that's my own that's my own thinking after a long time in a lab, and I hope to have a lot more days to spend in it.
Well, I appreciate that thinking, and appreciate, and for everyone that's going to be seeing the show, that you're there running Wistar in your lab and overseeing everybody else, because you need to be fighting this fight, and it's an important fight for us to win. So once again, thank you very much for spending the time with us today.
My pleasure, Ira.
Dario, it was a pleasure meeting you.
Likewise. Thanks so much.